All right, so chapter 42 is going to be split up over two videos because if I do all this at once, y'all are going to be very unhappy with me. So we're looking at circulation and we're going to look at gas exchange. We need to have ways for organisms to be able to exchange materials with their environment. Those exchanges are going to need to take place at the cellular level. We need materials to be able to cross the plasma membrane. When you have a unicellular organism, it actually is able to do it directly with the environment. But for multicellular organisms, for the most part, you're not going to be able to do that. Gills would be one example of an exchange system that's a little more specialized because oxygen moves from the water directly into the blood vessels or diffuses, and then carbon dioxide diffuses back in the opposite direction. How the transport things are transported within organisms and gases are exchanged is going to have a lot to do with how those organisms function every day. Okay, so circulatory systems are able to kind of connect all the different cells and tissues throughout the body to one another. Diffusion is going to play a big role. How long things diffuse for is going to be dependent on the distance that they're traveling. It works a lot better if it does it over small distances. So that's why when you have your smaller animals, they can kind of do that more immediately with that surrounding medium. But for larger animals, it's going to be more beneficial to use the circulatory system to make that happen. So, your circulatory system will have your circulatory fluid, vessels that connect with one another, and then some sort of muscular pump, most often called the heart. So, it connects the fluid that's going to surround your cells, that interstitial fluid, with all the organs that are working together so that gases can get exchanged, nutrients get where they need to go, and waste materials are removed. You can have both open and closed circulatory systems. You can also vary in what types of circuits and how many circuits you have. But the point behind a circulatory system is to help minimize that diffusion distance. So, some examples of open circulatory system organisms. You talked about this a little bit in your diversity unit: insects, arthropods, mollusks. The blood is able to bathe those organs directly. So, there actually isn't really blood in those particular organisms. The blood and the interstitial fluid are kind of mixed together, and we call that hemolymph. In a closed circulatory system, it is separate from the interstitial fluid, the blood is. And so that's going to be more effective at getting the fluids to those tissues and cells because you're not going to have oxygenated and deoxygenated mixed all together. So, examples of closed circulatory system organisms: your annelids, your cephalopods, and your vertebrates. Okay, so those are just kind of showing you a breakdown as to what the difference between an open and a closed circulatory system. Humans are part of the vertebrates. So we have a closed circulatory system: heart, blood vessels, and blood. Um, Vertebrate hearts can vary. They can have one or two atria, they can have one or two ventricles. Um, Blood will enter through atriums and it leaves through the ventricles. Skip some. I feel like I did. Okay, so that was a heart. Sorry. Blood vessels. Three main types of blood vessels: your arteries, your veins, your capillaries. Arterioles would be a smaller form of arteries. Venules would be a smaller form of veins. Blood only moves in one direction through these. These are all one-way streets. Um, it's not determined by the oxygen content, it's literally determined by the direction it's moving. The arteries are going to take blood away from the heart, while the veins are going to return blood to the heart. So, as I said before, arteries are going to turn into arterioles and they will eventually get the blood to your capillaries. The capillaries are where we have the really thin layers of cells. That's where we have the chemical exchange, the gas exchange between the blood and the interstitial fluid. And then once that exchange has taken place and the blood leaves the capillary beds on the venule side, the venules will come into veins and then they will return the blood back to the heart. In a single circulation system, examples would be your bony fishes, your rays, and your sharks. They have just a two-chamber heart. The blood goes through the heart. We're not trying to separate oxygenated from deoxygenated, and it goes through the two capillary beds before it returns. I'll show you a picture of that in a second. Double circulation would be separating the oxygen poor and the oxygen-rich blood. Um, in both reptiles and mammals, the oxygen poor blood goes through a pulmonary circuit to get oxygen from the lungs. Amphibians, they're not just going to exist on land, they can also survive in the water. So they have a pulmocutaneous circuit where they can take up oxygen not just through their lungs, but also through their skin. And then once the blood has been oxygenated, it's able to be delivered through the systemic circuit. This is able to keep the blood pressure higher. So, depending on the needs of those organisms, adaptations may have come over time to generate or to have this type of system be present for different species. So there you can see your single species. You can see your oxygen poor blood and leaving, going into the artery, going across the gill capillaries. So it's getting its oxygenated, then it's moving straight to the body capillaries, and then it's coming back out of the body capillaries from the veins back into the ventricle. So you never have oxygenated blood move through that heart, other than the blood that's needed to keep the heart pumping. But the actual blood moving through that muscle is not going to be oxygenated. In a double circulation system, you have your deoxygenated blood, excuse me, leave the right ventricle. It's going to move up through the aorta into, sorry, not the aorta, through the pulmonary artery into the lungs and the lung capillaries as it goes through that. So we're going to go from the arteries to the arterioles. We'll move into those capillaries. And then as we come out of the lungs and we've gotten oxygenated, we use the pulmonary vein to pump the blood back into the left atrium, help to get the pressure up. We're going to move it down through the left ventricle. Now it's going to leave through the aorta to feed the various organs throughout the body. As it goes through its gas exchange, the capillaries are going to send the blood back out with any of the waste materials, any of the carbon dioxide, and move it through the superior and inferior vena cava to go back into the right side of the heart via the right atrium. Okay. So amphibians have two atria and one ventricle. I think this is pretty wild. So the ventricle is able to pump blood into an artery that basically splits it between the pulmocutaneous circuit and the systemic circuit. Why this would be beneficial when they're underwater, that blood flow to the lungs is pretty much cut off. It's only going to get what it absolutely needs. Reptiles have two atria and one ventricle, such as turtles, snakes, and lizards. Notice birds are an exception up there. Alligators, caimans, crocodilians will have a septum distinguish, divide up the ventricle. And then reptiles will have double circulation. They'll have both the pulmonary circuit and the systemic circuit. So mammals and birds, why we dare to be different. We have two atrium and two ventricles, again, with the left side getting the oxygen-rich blood and the right side getting the oxygen-poor blood. We're endotherms. We have to make our own energy. So we need to have a more efficient way of getting our blood circulated throughout our body so that we can get the oxygen we need to the cells so we can make our ATP and generate our energy. Okay. So those are some comparisons between the different systems we just kind of talked about. Focus more on how hearts actually are able to contract in double circulation systems for mammals. Basically, we need oxygen all the time. And so the heart is our means of making that happen. The mammalian heart has that pericardium layer on the top of it. And it's going to have a couple of thin walled atria and then some thicker ventricles. And basically, if you clench your fist, you have the size of a heart. So I went through this with you on that previous slide. You're welcome to read through it again, but it's just talking about the pathway blood has to travel to become oxygenated, to reach the various cells throughout the whole body in terms of organs and tissues, and then to return back. Okay, so those are just showing you again, blue is where the blood is deoxygenated, red is where the blood is oxygenated. So we can see that the blood is coming in at the right atrium. It's exiting the right ventricle. It's going up to get oxygenated. It's coming back in through the pulmonary vein. As it leaves the left atrium and left ventricle, then it's able to go out through the aorta. Okay. And the deoxygenated blood again leaves initially through the pulmonary artery. The heart itself needs blood as well. And so the blood that we've been talking about passing through the heart is not directly going to go into the heart's muscles. It is able to enter the heart tissue, the cardiac muscle, via the aorta. And those are those coronary arteries that you're always hearing about. That if people have heart problems, that's where the issues typically are going to arise. The heart contracts and relaxes in what's called a cardiac cycle. When it's contracting, you have your systole taking place. When it's relaxing or filling up, that's your diastole. So contracting would be your pumping. Your heart rate is how often your heart is beating going through that cycle, how many beats per minute. The volume is how much blood it's able to take in, and then the output would be a combination of those two. The volume of the blood taken in per minute. So it would depend on both your heart rate and your stroke volume. Okay. There are some valves, kind of like we talked about with the digestion. There are, as opposed to sphincters, there are valves that kind of help to make sure that there's no backflow. You have your atrioventricular valves, which separate the atriums and ventricles from one another. And then you have your semilunar valves, which are going to help to control the blood moving into the aorta after it's been oxygenated and moving into the pulmonary artery on its way to be oxygenated. And that's the lub dub sound you hear when you have a heartbeat. The lub would be the AV valve, and the dub would be the semilunar valve. If there is blood moving backwards, because there's some sort of valve issue, depending on the significance of it, will have a role as to what happens. That's when you have a heart murmur. Why a heart can beat without blood moving into it has to do with your pacemaker, your sinoatrial node. This is able to be excited by itself. It does not require any nervous system signal to make it work. And this determines basically when those cardiac muscles are going to contract. Um, so this is what gets things started. Again, it's regulated by both your sympathetic and parasympathetic nervous system. We'll talk about those in another chapter. Sympathetic helps to move it up. So if you're running around and you need it to pump more blood more quickly, your sympathetic division would help to encourage that SA node to move things along. And if you're relaxing, like you're trying to go to sleep, the parasympathetic division would help to slow it down. Hormones will play a role, as will temperature. As the SA node is signaling, it will send impulses to another node, the AV node. Those impulses will be delayed just a little bit. They'll move to these fibers that are kind of throughout the ventricles and the Purkinje fibers, and is when the ventricles will actually physically contract. For the atrium to work in concert with one another, the left and right, and for the ventricles to work in concert with one another, since they're carrying totally different types of blood in terms of oxygenated and deoxygenated. Those gap junctions we talked about in terms of how cells are connected to one another are how they're going to communicate to know that they need to work in concert with one another. So blood pressure and how blood pressure kind of affects your blood vessels, how things are arranged. how water is able to move in plumbing systems is gonna it's kind of a weird analogy, but it's similar to what's got to happen with circulatory systems for animals. Okay. Blood vessels are going to have a cavity, which is referred to as the central lumen. The initial layer of cells surrounding it is the endothelium. That helps to kind of minimize some resistance. There's not a whole lot there. Your capillaries have really thin walls. They just have this endothelial layer and the basal lamina, which is the extracellular matrix that they actually make. And that's where the exchange of materials is going to take place. Um, so they don't have to move as much blood through them. They actually want the blood to stay on them a little bit longer. Arteries and veins have got to do a little bit more work. So they're going to have some other types of cells besides the endothelium. They're going to have smooth muscle and they're going to have connective tissue. The arteries are going to have a little bit thicker walls because, again, they are the ones taking the blood straight from the heart. And especially on the oxygenated side, they've got with that blood having the oxygen there, you've got the pressure even built up more. The veins are going to be able to, they've got to somehow get the blood back up to the heart. Um, the ones that are going all the way down your legs and into your stomach and things like that. And so muscles are going to be what help to move that blood back upwards against gravity. Okay, so there's some pictures of what we're looking at in terms of the artery versus the veins. And then you've got your arterioles, your venules, and your capillaries. So again, arteries, the blood's coming straight from the heart. The veins are taking the blood back up. So the veins are not going to be taking up as highly pressured of your blood. So they have a larger volume, where the arteries are going to have a little bit smaller volume. They're going to have a higher pressure. That goes back to kind of Boyle's law. Gotta love chemistry. Okay. Blood flow velocity. So laws of governing the movement of fluids through pipes will affect blood flow and blood pressure. Velocity, the speed at which the blood is moving, is going to be slowest in the capillary beds. You've got a larger surface area, a little bit more resistance there, and we actually want it to be slower. This is a good thing because this is where you're going to be having those gases exchanged and the other substances that are being carried to the blood that the cells and tissues need. Okay. So again, you've kind of got, as you move from your arteries all the way through your to your veins, you can see that the area is going to be greatest at the capillaries, and that's where the speed is going to kind of drop off. We're going to see that the smallest volume is with our arteries, and that's where our pressure is going to be the greatest because we've got the greatest we've got a smaller volume, so our pressure is going to build up. And as our volume increases, we're going to see the pressure is going to diminish. So, kind of like diffusion, blood's always going to move from higher to lower areas. The pressure, when we measure blood pressure, you're measuring how much the blood is pushing against the vessel of your wall. If you have a rigid vessel, your blood pressure is going to stay pretty consistent. So that would be like, um, I'm trying to think of a good example for that. That would be like having a water bottle that's made of a pretty sturdy plastic. It's going to be able to keep that shape, as opposed to one that you might spend like $1 on at a 7-Eleven. It's not going to be able to keep its shape as much as the water levels decrease. So it's not as rigid. The pressure isn't going to be able to stay as constant. Systolic pressure is the pressure in the arteries when the ventricles are contracting, ventricular systole. As you saw in the picture before, that's when you have your highest pressure. The diastolic is the pressure when they are filling up, and so it'll be much lower than your systolic. And then when you are having your pulse measured, it's just basically how often those artery walls are moving. It's not really telling you anything about what the pressure's like in there. It's just telling you how often those walls are being pushed against one another with your heartbeat. So we can measure blood pressure by, blood pressure is determined based on how well your heart is working and how things are moving through your arterioles. How things are, what type of resistance is the blood encountering as it's moving through them. Vasoconstriction is when the smooth muscle in the arterial walls is contracting, so that's going to cause your blood pressure to rise. That's what happens when you take things like Sudafed, that pseudoephedrine, it will cause vasoconstriction to take place. Vasodilation is causing those muscles to relax, and that's going to reduce the blood pressure that exists. So you would take a vasodilator, would help your muscles not to contract as often, and that would help to make the blood pressure decrease. So, depending on, and we'll talk about this a little bit later, if you have blood pressure issues, let's say you have really low blood pressure, you might be asked to take a vasoconstrictor to kind of help that out. Or if you have high blood pressure, you might be asked to take a vasodilator to help to get those smooth muscles to chill out a little bit. Um, so, these sometimes your body may need to have a higher blood pressure, and other times it may need to have a lower blood pressure. And there are chemical substances that are made by your body that can help with this in over periods of time. Nitric oxide, it's not laughing gas, can promote vasodilation. And endothelium, which is made by endothelial cells, can promote vasoconstriction. Typically, when you measure blood pressure, you want to measure it for an artery that's at the same height as your heart. Um, a healthy blood pressure, this says 120 over 70, 120 for your systole, 70 for your diastole. And I would say whether you're 20 years old or 50 years old, that's usually the goal, anywhere from 110 to 120, 70 to 80. If you go through fainting, you're not getting enough blood to your head, so you've got a lower blood pressure than what you need. If you're something like a giraffe and you have a really long neck and you really need to push the blood up to your head, you're going to need a higher systolic pressure because you've got to really fight gravity to get that blood up there. Again, we talked about with veins that the blood is not going to have that pressure to help move it through. So it's going to use the smooth muscles that are present as well as the skeletal muscles surrounding the blood. And then when you take in air, your vena cava will expand, and that will help to get more blood up into the vena cava so that you can get it into your right atrium. And there are valves in the vein too that help to prevent the blood from going in the wrong direction. Okay, so this is just showing a picture of how blood pressure is taken, why they want to make it super tight to get started. They want to kind of cut off the blood from moving. And then when they first start hearing the blood move through at that minimum level, that's when you're going to get your systole, when you first hear the contractions. And then when you no longer are hearing the contractions, that's when you're going to get your diastole because now the blood's just filling up. Okay. So again, blood's always going to move in one direction, atriums to ventricle, or sorry, atriums to veins. Okay. Capillary function. Not all of the capillaries in your body are going to have blood going through them at any given time. Major organs are going to keep those capillaries working, aka your heart. But depending on what your body needs, you're not necessarily going to have all the capillaries in your body filled with blood all the time. Mechanisms that will regulate this distribution. Contraction of your smooth muscle layer in the arteriole, and I'll show you a picture of this, along with precapillary sphincters, which help to kind of determine when the blood should move between the arterioles and the venules. Some other factors that can play a role, your nerves, hormones, and then other chemicals that might be taken. The exchange is going to take place between the thin endothelial walls of the capillaries. So it's blood pressure difference, or the difference between your blood pressure and the osmotic pressure of that interstitial fluid that helps to determine whether fluid is going to come out of the capillaries on the arterial side of things and to go into the capillaries on the venule sides. Um, Blood proteins, for the most part, are not going to move into those endothelial cells, and blood cells are not going to move into those endothelial cells. They're way too big. Okay, so I told you I would show you a picture of those sphincters kind of contracting and relaxing to determine whether the blood should move through or not. So you can see when they're contracted, it's not going through the capillaries as much. And then when they are, or sorry, when they're relaxed, it's moving through them. When they're contracted, not so much. So remember that we want to make sure that your cells are getting the nutrients that they need, or the molecules, the hormones that they need to be able to do what they need to do. And so blood pressure is going to be much higher on the capillary end, and that's going to cause the osmotic pressure to be a little bit lower. And then as we continue to move through the blood pressure, as we are exchanging the oxygen and everything else, we see that the blood pressure and the osmotic pressure are getting closer and closer to one another. Sometimes there is fluid that leaks out from the capillaries, and that fluid gets returned via the lymphatic system. This fluid's called your lymphs, or called lymph, and it's going to enter on the venous side of the capillary bed. And there are veins for the lymphatic system that will, or sorry, the lymph will drain into veins in the neck. And there's, again, that we've talked a lot about checks and balances so far with the veins and with the arteries. There are valves to prevent that backflow. But the lymph is throughout the whole body. And so the lymph nodes that we are going to get the lymph to will filter it, and they help to protect the body against potential invaders. Sometimes when you have had issues with lymph, like disruptions in making that lymph move, that's when you will have some swelling, edema. All right, so the components of blood are going to play a big role in determining how things are going to be exchanged, how we're able to transport, and how we're able to protect the body. In open circulation, everything comes into contact with everything. In closed circulation, the blood is going to act, the connective tissue that's going to be able to connect your organs and various tissues um, to the components they need from via the oxygen or from food materials you've digested that organs would not normally encounter with each other. Okay. Your blood is made up primarily of plasma. 55% of the volume of your blood is that. And that's going to contain a lot of water, but also ions and some plasma proteins, whatever is trying to be transported at the time. It can have an impact on your pH, your pressure, your viscosity, how thick it is, and how well it's able to move transport. So, this is why when you get blood work done, they can find out a lot of information as to what's going on. Your plasma contains a lot of key components that give you an idea as to how healthy your body is. And then other components of your blood will help them determine things such as your erythrocytes and leukocytes, as well as your platelets. And we'll spend more time on those two types of cells that are suspended in your blood plasma. And then we're going to spend a little bit more time talking about platelets. Okay, so you're welcome to go back and take a look at that. But it just summarizes what we talked about now and what we're getting ready to talk about. Again, it's kind of giving you on the right side there what you would expect to see. And those numbers give, again, your medical professionals a better idea of the healthiness of what's going on within your body. So, erythrocytes are what are the most likely blood cell you're going to find. They're the ones that contain your hemoglobin, which can allow the oxygen to move. Each hemoglobin can bind 4 O2 molecules or up to 4 O2 molecules. Um, Red blood cells do not have nuclei. They do not have mitochondria. They get used up. And we've talked a lot about sickle cell before. Sickle cell is when you have the abnormal hemoglobin proteins, and when they are not in the proper shape, they can actually cause blockages in blood vessels, or they could rupture them. And that's when you will have, if you know someone who has sickle cell, you may hear that they talk about being in pain sometimes. This is why. Leukocytes, your white blood cells, monocytes, neutrophils, basophils, eosinophils, and lymphocytes. These help to break down the bad things that enter, aka bacteria, or just generic debris, things that have started to break down. They also help to make antibodies. We'll spend a little bit more time talking about that, I believe, in our next chapter. They are not just found in the circulatory system, but they are found in other places as well. And then we have our platelets. Platelets aren't true cells, but they are really super important because they help to clot. If you have had the blood that's not staying where it needs to be, maybe you've had an injury. Um, so, coagulation will help to form one of these solid clots. It's taking fibrinogen, which is inactive, turning it into fibrin, which kind of helps to connect everything together to kind of make a plug. Um, and that's all good and well if you have an injury or something that you need to heal. Where we don't want these blood clots to form are within your blood vessels, because then those vessels can travel to your arteries and or to your aorta and cause a lot of trouble. So they can form thrombosis, and we don't want those. Okay, so clotting factors can come from your platelets, cells that are damaged, or from your plasma. And again, they can kind of help to block things off. And I remember I had to do a paper about this in college, where enzymes help to break things down to cause this cascade effect to take place. Where we don't want one of these to happen, again, is in your veins. Um, Stem cells. Again, I talked about how the blood cellular elements are always wearing out, especially your red blood cells. But your white blood cells and your platelets can also wear out as well. Those are made from your stem cells in your bones, ribs, vertebrae, sternum, pelvis. EPO, you may have heard about that in terms of athletes injecting that when they are not supposed to. Helps to encourage the production of those red blood cells. Um, and there are certain amounts of it that we obviously need if we're not getting enough oxygen throughout our body, but we definitely don't want it to be to have too many red blood cells wandering around our body. That's going to cause you to have a greater amount of oxygen being generated, allow you to make more energy, which is why athletes can get in trouble for it. Okay. We're supposed to have what we need. So stem cells, you've got your lymphoid stem cells, your myeloid stem cells, and that's how we get um, our lymphocytes from the lymphoid stem cells, and we get the red blood cells, the erythrocytes, and the other leukocytes, and the platelets from the myeloid stem cells. So this will be the last part of this video. Cardiovascular disease. Heart disorders, blood vessel disorders, these account for the majority of the deaths in the United States. Um, Atherosclerosis, heart attacks, strokes, uh, heart disease risk increases when you have a much higher LDL to HDL ratio. You want to have, remember we talked about cholesterol back in the day, how that's going to help to keep your membranes fluid. LDLs help to bring cholesterol to make more membranes. So we like that type of cholesterol. HDL takes the cholesterol out and takes it back to the liver. So you can have a great LDL number, but if your HDL number is too high, that still is a problem. So you want your LDL to be um, to be higher compared to your HDL. And doctors will talk to you about whatever the heck that is, especially as you get older. Inflammation can also play a role in impacting with atherosclerosis. What the heck is atherosclerosis? That's when you get the plaque deposits in your arteries. And if you have a heart attack or a myocardial infarction, you've had cardiac muscle die off. And that's happened because one of the coronary arteries that's giving the heart the oxygen rich blood it needs to be able to do its job isn't getting the blood to the heart muscle. And if the heart muscle isn't getting the blood, the heart has to keep working. If it's not getting what it needs, it can't do what it needs to do, then it's just going to stop working. Angina pectoris is partial blockage of those, causes chest pains. And then strokes are also dealing with artery blockages, but they are due to nervous tissue in the brain typically dying off, whether there's been some sort of rupture or blockage of arteries in your head. Okay, so when you start to develop some of these plaques or atherosclerosis, you've got your LDLs floating around, and then you've got your macrophages trying to chew up these LDLs, and they form these foam cells. And you start to see some of the, I want to say it's the endothelium. Um, Actually, I think it's smooth muscle, kind of help to build out, and I think that's in part two. And you've got some more of that extracellular matrix, so it's becoming a more permanent spot, plaque. Um, you eventually will cause those foam cells to break down, and then you'll have more of a specific cap over it. Um, that'll cause the plaque to rupture, and when that plaque ruptures, that can cause more to build up on top of it. And as you can see, the space in that artery is narrowing, and the artery was already pretty narrow to begin with. And if you're trying to push a lot of blood through an artery and there's not enough space to push it through, we've got a problem. So, factors that are of concern when it comes to cardiovascular disease. Obviously, that ratio of LDL to HDL. And then we're going to talk about ways you can possibly treat these things. Exercise, um, not smoking, um, trying to eat a less fatty diet, especially one with trans fats. Those are the ones with the bad lipoproteins. Taking statins, inflammation, again, can have a role with atherosclerosis, but it also can be a factor in thrombus formation. So aspirins can kind of help to do that. Again, don't just take an aspirin a day. Definitely talk to your doctor. Hypertension, having higher blood pressure, because that's kind of an indication you've got a smaller volume going along. That will have an influence on atherosclerosis, heart attack, and stroke. Ways you can work to treat that, obviously medication, exercise, as well as dietary changes.